Hi, welcome to the screencast. This is just a very brief overview of organelles. I don't consider them super important for this class, so we'll be kind of quick. Just to refresh your memory, you have your cell membrane. In the middle of the cell is the nucleus, and then all this stuff, yellow, is cytoplasm, and then floating around in the cytoplasm are organelles, or what can be called little organs. And they do kind of function like organs in your body in that each one performs a different function or set of functions for the cell. You know, the way your kidneys do something and your liver does something and all that. So I'm going to cover them in a specific order where we're going to talk about how proteins are made and then how they end up where they're supposed to be in the cell. And then we'll go on to some organelles that don't have anything to do with proteins. And I made this in part because there are certain terms that get associated with certain organelles that I do not like. They are too easily misused, so I have inserted what I think is more scientifically accurate and specific terminology. And whoops. There we go. So first up then is the nucleus. That is the thing in the middle of the cell. It is bound by a nuclear envelope. The envelope has pores in it, but that is not going to be super important for us. But just so you know, that is how larger particles can be transported into and out of the nucleus. I am going to want you to know that what is in the nucleus is a substance we call chromatin. Chromatin is DNA, which you probably already guessed is in the nucleus. But the DNA molecules themselves are very long, very thin, kind of like pasta, very easy to get them all tangled up and have them break because of shearing forces or different mechanical forces. So your cells wrap them around proteins like you would wrap thread around a spool to keep all your different color threads organized. So that combination of DNA plus the proteins that it's wrapped around to manage it is called chromatin, which just means dark stuff, dark matter. Inside of the nucleus is a neighborhood called the nucleolus. This is where ribosomes are going to be manufactured. You are going to see that a ribosome is a combination of protein and RNA. The RNA is made in the nucleolus, so they assemble at least some of it, your cells, inside the nucleus and then export the ribosomes through these nuclear pores. So then, right, the reason why you have a nucleus is the nucleus stores DNA. The DNA encodes proteins. So every time you need to make a protein, you take your message from your DNA, you take the information encoded in a gene and transcribe it into an mRNA message that is then shipped out to the cytoplasm, where there is a ribosome waiting for it somewhere. Let me just skip ahead one real quick. So you will notice some ribosomes are just floating around freely in the cytoplasm. Those are the ribosomes we're talking about right now. So if a protein is going to do its job just floating around in the cytoplasm, that is its final destination, its location, then it will be synthesized by a ribosome floating around in the cytoplasm. All proteins are synthesized by ribosomes. So the ribosome is the only organelle whose job is protein synthesis. So there it is there. And it is RNA plus proteins, but I'm probably not going to ask you about that. You do want to keep in mind that it can be floating within the cytoplasm or, I should say bound to endoplasmic reticulum. So this then gets us to what is called the endomembrane system. That is not an important term, but I had to give the slide a title, so that's the term it gets. You just need to know that all of the organelles that are bound by a membrane, so the ones we're about to cover, it's all phospholipid bilayer. So the cell membrane, all of your organelles, and these little blobs here called vesicles those are all made out of phospholipid. So phospholipid is used to package things, and then that same material can be used to transport large bundles of proteins and other polar molecules within the cell and move them around from one location in the cell to the next. So we're going to be talking about our endoplasmic reticulum, specifically the rough endoplasmic reticulum, the Golgi, and some vesicles. So first up is, well, ER is just endoplasmic reticulum. I'm not going to spend too much time talking about what that means. Reticular means branching network. Endoplasmic means in the cytoplasm. So this is a branching network of tubes or tubules that is in the cytoplasm. It is going to do a couple of things for us throughout the semester. We are going to think, sorry, I got ahead of myself. The first type of endoplasmic reticulum we are going to talk about is rough endoplasmic reticulum. It is called rough because it has ribosomes on the surface. So the ribosomes are going to be synthesizing the proteins. They get fed into, like squirted into the endoplasmic reticulum by the ribosomes as they're being made. And then the ribosomes are going to process the proteins. We do not have time to talk about what kind of modifications are made to the proteins in the ER, but just know that there is some processing that takes place. And then those proteins are going to be shipped to another part of the endomembrane system. So rough ER is protein processing and the first step in routing proteins towards their final destination within the cell. So this is for proteins that need to end up somewhere specifically, like in a mitochondria or a lysosome or outside the cell. If a protein is not going to just float around in the cytoplasm, it's made by a ribosome on the ER, so this rough ER. So those proteins, here we go, this green thing is a protein manufactured by a ribosome in the rough endoplasmic reticulum. It gets dumped into a little vesicle, a bubble of membrane, which again, it's kind of like a soap bubble, that is going to transport that protein to the Golgi. The Golgi is going to do some more processing, but we don't really care about that. We are going to think of it as being what packages proteins so that they ultimately end up where they're supposed to be, whether that's somewhere inside the cell. So maybe there's a protein that's going to be shipped off to a lysosome or the mitochondria, or outside the cell, either on the surface of the cell membrane or just dumped out into the extracellular fluid the way a neuron might be. So we're going to say Golgi, packaging, and secretion would be a good word to put in there. This says shipping off, but I'm going to change it right during the middle of this. Right during the middle of the screencast, and I'm not going to go back and edit this thing because I'm just trying to get it done. That's Golgi. Then there is smooth ER. So we're done talking about how proteins get made and where they end up where they need to be. There is endoplasmic reticulum, like over here, that doesn't have ribosomes on it. That is called smooth endoplasmic reticulum. We are going to think of it primarily as synthesizing lipids, so steroid hormones and phospholipids. Like just think as a cell grows, it's going to need a bigger membrane. The phospholipids that get dumped into the membrane are going to come from the smooth endoplasmic reticulum, as well as steroid hormones like estrogen and testosterone. In addition, there are certain metabolic byproducts that accumulate inside of cells. Your body makes them. They are waste. Some waste you can just excrete as is. Other waste you want to process so that it is less toxic to the cell. Think like the catalytic converter on your car, making your exhaust less toxic. So there are a number of metabolic byproducts that are processed in the smooth ER to make them less toxic, and then presumably they're just going to be excreted. Peroxisomes do something that sounds very similar. So they're just the small circular organelle that doesn't look like much under the microscope, and they are also going to process metabolic byproducts. They're called peroxisomes because what they do is sort of deactivate what are called reactive oxygen species like hydrogen peroxide, which can damage things like DNA. So somewhere inside of the peroxisome is something that the hydrogen peroxide can react with, and then it's going to get turned into water and something else. So whatever is in the peroxisomes is something that can be damaged and then gotten rid of. So dangerous, damaging, highly reactive chemicals are allowed to just do what they do in the peroxisome. So it's sort of like where the police would take a bomb to detonate it so it doesn't hurt anybody. That's the peroxisome. Again, processing metabolic byproducts. You will see the word detoxification used a lot, which is not necessarily a misuse, because these metabolic byproducts will become toxic if they're allowed to accumulate. However, people also try and sell you like detox tea and other detoxification products. My understanding is that most of those don't do anything, and so I avoid using the word detox. If you enjoy an herbal detox tea or any detoxification product and you feel better after using it, please continue to use it. If you're thinking about whether or not you want to buy it, I would say save your money, but, maybe do a little research, look it up. How many do we have? We're almost done and I have 4 minutes. Mitochondria. They're usually called sometimes the powerhouse of the cell. This slide calls them the energy transformer. I'm just going to say they produce ATP. And by transformer, that means that different kinds of compounds, like sugars or little chunks of sugar and deaminated amino acids, those get dumped into the mitochondria. The mitochondria process them, harvest the energy from those, and then use that energy to produce ATP. So they're transforming the energy, so to speak, because you can't really transform energy. You can just transfer it from one molecule to another. And so they're taking the energy from all of the types of nutrients that you eat and using those nutrients to make ATP. The cytoskeleton we're going to do quickly. There is only one cytoskeletal protein that I'm going to ask you about, and that is called microtubules. Those are these green things here. We are generally only going to see and talk about microtubules during cell division, but as you can see, that green is there in between cell divisions during interphase. And it plays an important role in transporting things around inside the cell. So those vesicles that we were talking about that get moved from point A to point B get basically clipped onto the microtubule like it's some sort of cable, and then they zip line from one organelle to another. I think this is our last. Yes, and we're going to come in under 15 minutes. These are the centrioles. There are 2 of them. They're held together by some protein goop. So that whole structure with the 2 of them plus the goop is called the centrosome. I don't care if you say centrioles or centrosome. I don't differentiate between the 2. What they do is organize or direct the formation of microtubules. So they control your microtubules. During interphase, the microtubules make up a transport network, as I was talking about before, and then during mitosis, they form what is called the mitotic spindle, which is what is going to pull the replicated chromosomes towards either pole of the dividing cell, ensuring that each new cell gets 1 copy of all of the original chromosomes. There was one other point I wanted to make, and I've forgotten what it is. And I'm going to stall for like 10 seconds and see if I can remember the point. Oh yeah, these also get replicated before mitosis. Not an important point, but somebody noticed that there was only 1 centrosome in the cell. So that replicates before you go into mitosis, and then you have 2. All right, that's it. 15 minutes, hopefully not too long. Thank you for sticking with me this far.